So now we are going to discuss, previously we discussed about the proton accumulation in the lumen.
Okay.
So now we are going to discuss about how this gradient, this proton gradient is break. How this proton gradient is broken down, we are going to see. Complex structure broken down into simple. Which complex structure?
Complex.
Proton actually accumulates inside the lumen. Okay. So now the concentration of proton is more in the lumen than the stroma. Lumen of thyroid than the stroma. Okay. So this proton gradient, this proton concentration which is high here and low in the stroma, it creates a proton gradient.
Okay.
So with this gradient ATP are formed. Okay, high energy ATP bonds are produced with this gradient. So how it is produced, what mechanism it is produced through by using these protons we are going to see here. So this breakdown of gradient, it releases energy. When this proton gradient, that is the concentration of proton which is higher inside the lumen, so when that concentration is high, when it equalizes or when it becomes less, then it pays to the release of energy. Okay. So we already saw that the dynamic photosystem 2 is here and electron carrier, and photosystem 1 is here, and if this is the thyroid membrane, so here if this is the thyroid membrane, so here we saw that F0 and F1 are present. Okay. So this two are important to produce ATP. So these two acts as ATPase. Okay. So here this movement of protons, okay, across the membrane through the transmembrane channel. Transmembrane channels. This one is F0. We have already seen in the last video F0, and this one is the F1. Okay. So this across the membrane through the transmembrane channel. Transmembrane channel is nothing but F0 because it lies between the two membranes. Okay, in between the two membranes it lies. So transmembrane channel of the F0 of the ATPase. Okay. So ATPase is of two kinds. First one is F0 and the second one is F1. So F0 is embedded between the two membrane and F1 is outside facing the stroma. It is attached to the F0 and it is facing the stroma, this F1. Okay. So the smaller is known as F1. So what does F0 do? F0 initiates the facilitated diffusion of these protons inside the lumen to the outside stroma. Okay, it facilitates the movement of them. So here after that, after the movement of these protons through the F0, there comes F1. So this F1 synthesizes several molecules of ATP. Okay, several molecules of energy packed ATP. Okay. So this is chemiosmosis. Okay. So what are the requirements of chemiosmosis to occur?
ATP and NAD.
ATP is produced. Okay. So proton gradient, membrane, proton pump and ATPase. So these are required for chemiosmosis to occur. So it produces ATP and NADPH. So this is overall light reaction where through light it produces ATP and NADPH. So now in dark reaction, these ATP and NADPH are used in dark reaction. Okay. So this ATP and NADPH is used for fixing carbon dioxide and synthesis of sugars. So these are known as dark reaction, the use of ATP and NADPH.
So we saw light reaction and dark reaction.
Yes. Both are no. Next only we are going to see about dark reaction. Only now we have seen only light reaction, how ATP is synthesized. Okay. So next we will see about where are the ATP and NADPH are used. Okay. We are going to the seventh topic, which is where are ATP and NADPH used. So we are going to see the ATP and NADPH which are produced in the right reaction that are used in the dark reaction. So we are going to see where are these ATP and NADPH used.
In dark reaction, where it is used.
Yeah.
Okay.
So now dark reactions.
We are going to see about dark reaction.
So this phase is also known as biosynthetic phase because of the production of sucrose or starch with the help of carbon dioxide fixation. Okay. So here there are two cycles. Okay, mainly in all.
Calvin cycle.
Yeah, the Calvin cycle is also known as C3 pathway.
Okay.
And the second one is C4 pathway. Okay. So C3 pathway was first discovered by Melvin Calvin, and hence it is known as Calvin cycle. Okay. So why it is C3 pathway and this one is C4 pathway?
No.
So it is because after the fixation of CO2, carbon dioxide is fixed in all plants, right? So after the carbon dioxide fixation, the first product formed is 3 carbon product.
So that the 3 carbon they put there and the carbon they put. So carbon.
C3 3 carbon product because 3 carbons. We found here
C3 carbon.
Yes.
Carbon.
So C3 pathway. And the first product was named as 3-phosphoglyceric acid. It was 3-phosphoglyceric acid. In short, it is PGA, phosphoglyceric acid. So the next pathway is C4 pathway. So C4 pathway, it is named as C4 pathway because after the fixation of CO2, the first product which is formed is oxaloacetic acid.
Oxaloacetic acid, okay.
Which is a 4 carbon product.
4 carbon product, okay.
So therefore it is known as C4 pathway. Okay, oxaloacetic acid is also known as OAA. So remember in Calvin cycle, the first product formed is 3-phosphoglyceric acid. And C4 pathway, the first product formed is oxaloacetic acid. Okay. So we are going to see the primary acceptor of CO2. It is fixed. This CO2 is fixed to something, fixed to some molecule, and it forms 3-phosphoglyceric acid. Okay. So what is this CO2 acceptor? What is accepting CO2 and becoming the product? So that one is ketose sugar, 5-carbon ketose sugar. It is a 5-carbon molecule. Okay. 5-carbon molecule accepts carbon dioxide which has 1 carbon and it becomes 6-carbon molecule. So this 6-carbon is separated into 2 3-carbon molecules. So that 3-carbon is 3-phosphoglyceric acid. Can you understand? So the primary acceptor of CO2 in C3 pathway is 5-carbon ketose sugar. So this ketose sugar is the acceptor of CO2 which contains 5 carbons in it. Okay. So next we will see the C3 cycle in detail. What is the C3 cycle and what is happening and how ATP and NADPH is used there we are going to see. Okay. So now we are going to see Calvin cycle. Okay. It is also known as C3 pathway. So the first fixation product here is
Calvin found that cycle.
Yeah. So what is the first fixation product here?
Ribulose
product in 3-phosphoglyceric acid, which is 3-carbon compound. So we saw that the first CO2 acceptor, it is accepting CO2. Okay. So it is a ketose sugar. That ketose sugar is ribulose 1,5-biphosphate. Okay. So this ribulose 1,5-biphosphate accepts CO2 and water molecule.
Okay.
So since it is fixing CO2, this process is known as carboxylation.
Okay.
So after this obtaining CO2 and water, okay, it forms the first product which is 3-phosphoglyceric acid. So which is 3-phosphoglyceric acid is also known as 3-phosphoglycerate, and this one is a 3-carbon compound. Okay. So after obtaining this, it undergoes reduction. Okay, reduction is that use of ATP, use of 2 molecules of ATP for 1 carbon.
Okay.
For 1 carbon, 2 ATP and 2
NADPH
NADPH.
for water.
No. Then it is also the same carbon. These energy are used. So we have seen that it is used, right? Where is ATP and NADPH used? So it is used in the dark reaction. In this reduction only it is used. So 2 ATP and 2 NADPH are used, and it produces 2 ADP, which is 2 ADP and 2 inorganic phosphate plus 2 NADPH.
Okay.
NADP condition. So for 1 carbon, 2 ADP and 2 NADP plus are released. Okay, here in this process. Okay, so after reduction, it produces triose phosphate. After reducing this one, it produces triose phosphate. From this triose phosphate only we get sugar and starch.
Sucrose and
Sugar, which is the sugar.
which is converted into
Yeah.
starch.
So this triose phosphate, after producing this, undergoes regeneration. Okay, in this regeneration process, it uses one ATP and releases one ADP for one carbon. So totally for one CO2, 3 ATPs are used to give 3 ADP, 2 ADP plus 1 ADP. Okay, so overall only 2 NADPH is converted into 2 NADPH. Whereas the ATP is 3 ATP is converted into 3 ADP. Okay. So after regeneration, again it regenerates. Again it regenerates ribose 1,5-biphosphate. So again the cycle continues. Okay. So therefore, totally how many carbon dioxide are going in to produce one glucose? 6 glucose is nothing but C6H12O6. Okay. Glucose is nothing but C6H12O6. So this is glucose. So 6 carbons we need to produce glucose. So therefore 6 carbon dioxide goes in. For 6 carbon dioxide, how many ATPs are required? For 1 carbon dioxide, 3 ATPs are used to produce 3 ADP, which releases to 3 ADP. Then for 6 carbon dioxide, how many ATPs are used? 18 ATP. So 3 into 6, which is 18 ATP. So how many NADPH are used for 1 carbon dioxide?
NADPH for NAD. How many NADPH are used for 1 carbon dioxide?
2 NADPH plus H is used and it releases 2 NADP plus. Okay. So here for the whole cycle only 2 NADPH is used. Okay. For 1 carbon dioxide. For 6 carbon dioxide it is 2 into 6, which is equal to 12 NADPH. So these are used. So 6 carbon dioxide is taken in, 18 ATP is taken in, and 12 NADPH is taken in. So what is the product?
NADP plus.
Yeah. So the product is 12 NADP plus. 1 glucose is produced. 1 glucose is produced. 18 ADP is formed and 12 NADP plus is formed. Okay. So this is the Calvin cycle. Okay. It regenerates the keto sugar, which is glucose 1,5-biphosphate, which is a 5 carbon sugar. Okay. So next we will see about C4 pathway, what is happening in C4 pathway. Okay.